Plasma membranes. So this is part of the transport across cell membranes topic, and you need to know the basic structure of all cell membranes, including cell surface membranes, and the membranes around the cell organelles of eukaryotes is the same. You need to know the arrangement and any movement of phospholipids, proteins, glycoproteins and glycolipids in the fluid mosaic model of membrane structure. Cholesterol may also be present in the cell membranes, where it restricts the movement of other molecules making up the membrane. So this is part of 3.2.3, the cells topic of lower 6th AQA, transport across cell membranes. So first of all, you probably know that the membrane is made up of a phospholipid bilayer, and the phospholipid bilayer has hydrophobic fatty acid tails which face away from water, and these phospholipids can move relative to each other, so they can swap places with each other, which is why it's known as a fluid model. And then every so often you've got some cholesterol which lies between the phospholipids in some membranes, and that provides stability. It stabilizes their position. So the fluid is from the moving part, the fact that the phospholipids move, as do the proteins around the membrane. And the mosaic is the fact that it's made of pieces like these proteins, glycoproteins, etc. are scattered in the bilayer. So phospholipids form a bilayer, and the hydrophilic phosphate heads face towards the intercellular fluid outside and the cytoplasm inside, because they like water, they face towards water. And you've got the fatty acids tails which point towards each other away from water because they are hydrophobic. Sometimes you've got some carbohydrates which are attached to a phospholipid, and they are called glycolipid. Glyco is from the glycogen of the carbohydrate attached to a lipid, and then a glycoprotein is a glycogen attached to a protein, or a carbohydrate attached to a protein. You've got some large proteins which span the whole membrane, which are called intrinsic proteins. So there's two sorts of proteins: large proteins that span the bilayer, and small proteins on each side of the bilayer, so they would be your extrinsic ones, but the ones that go through are your intrinsic ones. Proteins move around in the bilayer, which gives the mosaic appearance. So here you've got phospholipids, and then every so often you've got cholesterol. So fluid is from the fact that the phospholipids can move relative to each other, and the mosaic is that proteins are embedded and they vary in size and pattern like the tiles of a mosaic. So plasma membranes, in the phospholipid bilayer, the proteins are interspersed between the phospholipids, which is where you get the mosaic pattern from. So the phospholipid bilayer function is that it forms a barrier to water-soluble substances and ions, so charged particles. So it lets lipid-soluble things through, but not ones that are not lipid-soluble. So it does allow non-polar or lipid-soluble substances to pass. There are some polar molecules that can pass, such as small polar molecules like water. And another function of the phospholipid bilayer is it maintains a different environment on each side. So the functions of the membrane are they allow lipid-soluble substances to enter and leave the cell, they prevent water-soluble substances entering and leaving the cell, and they make the membrane flexible. So now we'll look at proteins in the phospholipid bilayer. So extrinsic proteins are ones found on the outside, like here, and they are used for cell recognition and also as receptors. So they can be binding sites for hormones or mechanical support. There's receptors, so some proteins are receptors, for example, hormones. Each receptor is specific to a particular substance. For instance, liver cells have receptors for the hormone insulin. Then you've got cell recognition. Some proteins are unique to that type of cell, and these proteins enable white blood cells to recognise foreign cells and our own. Glycoproteins and glycolipids. Glycoproteins, so sometimes you might see a protein like this in a membrane with phospholipids either side. So glycoproteins are attached to the membrane proteins, which are antigens, and they are used by cells to recognise other cells. White blood cell membrane receptors recognise self-antigens, and foreign antigens will incur an immune response. So intrinsic proteins are ones that span the phospholipid bilayer, and they are carriers to transport water-soluble materials across. And also they can be enzymes such as the ATP synthase enzyme, which is found in the inner mitochondrial membrane called the cristae. They can be channel proteins, and channel proteins form a pore through the membrane, and they let water-soluble molecules and ions through. So proteins have a hydrophilic channel which form pores that span the membrane, and it allows charged particles to pass through the hydrophobic centre of the phospholipid bilayer. You've got carrier proteins, and these have receptors which are specific to only one type of molecule, and they have a complementary structure or shape which can bind. So for example, hormones, drugs, viruses can bind to these receptors, but they have to have a specific receptor for a specific molecule, because the protein's tertiary structure is specific, which enables the molecule to bind. So molecules or messages are then transported into the cell. Another thing is they can allow large molecules to pass through the membrane, such as glucose, along their concentration gradient, and different carrier proteins facilitate the diffusion of different substances. As I said before, enzymes, some membrane proteins are enzymes. For example, ATP synthase, which synthesizes ATP, is found on the inner membrane of the mitochondria. So the functions of membrane proteins are, they are hormone and drug receptors, they are antigens, they can be involved in active transport and facilitated diffusion. Active transport would be carrier proteins, facilitated diffusion would be carrier and channel proteins. Enzymes such as ATP synthase, and they can act as structural support. So here's a summary diagram then. You've got your phospholipid bilayer with the hydrophilic phosphate head and the hydrophobic fatty acid tails. Just a bit of information, the bilayer is about 7 nanometers in diameter. You've got bits of cholesterol, which are for stability. You've got glycoproteins for recognition sites for chemicals like hormones. Glycolipids act as a recognition site for things like cholera toxins. You've got hydrophilic phosphate heads, we've said that. Protein molecules lying on the surface are called extrinsic proteins, whereas ones that span the bilayer are called intrinsic proteins. And now just a past exam question. Describe the fluid mosaic structure of a cell surface membrane. So as well as saying why it's fluid mosaic, you need to say about the structure. So they have phospholipids and proteins. There is a phospholipid bilayer. There is an arrangement of phospholipid molecules that go tail to tail. They are proteins which float and they can move in the membrane. There are intrinsic proteins which extend through the bilayer. There are extrinsic proteins in the outer layer. And there are channel proteins, glycoproteins, etc. And there is cholesterol.